Dr. Timothy Lee.
Hello. Today I'm going to share with you our research on the effects of Copaiba versus CBD oils on neuronal signaling pathway. Very quickly, please allow me to share with you what I do professionally. I run a molecular imaging and proteomic labs at the Rosemont University of Health Sciences. On a typical day at work, I tinker with ultra-fast lasers on a microscope system that I custom built. My associate, Dr. Yasuyo Urazaki, work on proteomic assays. So we use molecular imaging and proteomics to identify biomarkers for metabolic diseases and cancers. Since last year, we've been applying this technology to study the biological function of essential oils. The research that we presented to you on this very stage last year has now been published. In the journal Molecules. So in this study, we used proteomics to develop a physiological lie detector test to authenticate essential oils. This year, we used proteomics to study the effects of Copaiba versus CBD oils on neuronal signaling pathway. As we all know, both Copaiba and CBD oils might have the analgesic effects, but they might also have very different acting mechanisms. Because Copaiba interact directly with the CB2 receptor. In contrast, CBD oils does not interact with either CB1 or CB2 receptors. So the question that we want to ask is how Copaiba oils and CBD oils affect signaling activity within the neurons. So in this study, we used cultures of human neurons, and we treated them with Copaiba and CBD oils and measured the signaling activity over time. But we had a big problem. Which CBD oils should we purchase and use for this study? Now, choosing Copaiba oils is extremely easy. Because of the certified pure therapeutic grade, with guaranteed purity and quality of the oils. In contrast, there is no such standard for CBD oils. So we went to Amazon.com and we got thoroughly confused with the selections. Right? An average consumer would have a very difficult time to select the right CBD oils. Fortunately, we're not an average consumer. We happen to be the scientists who, together with dōTERRA, developed a physiological lie detector test that we presented to you last year. Right? So we said, bring them on. Bring on the challenge. We bought 6 CBD oils from 6 different vendors, and we put them through the test. Now, as it turned out, only 1 CBD oil passed the test and exhibits strong biological activities. The other 5 CBD oils failed the test, had no biological activity, or extremely low biological activities. So we called the company. We went to their website, to learn about the process by which they produce their CBD oils. And we learned that only one company sold us pure and unadulterated CBD oils that they collected right after CO2 extraction. The other 5 companies sold us adulterated oils that have been diluted with carrier oils and added with CBD isolates. So we chose the pure and unadulterated CBD oils that passed our test for this study. So how do we measure the signaling activity? Let's take a look at this signaling pathway, the PI3K, AKT, mTOR, which regulate neuronal growth, metabolism, and survival. To measure the signaling activity of this pathway, we measure the expression and phosphorylation level of 9 proteins which in the signaling cascade, as listed in this table. So here are some of the raw data that we collected. In blue are untreated control, in pink are treated for 30 minutes, and in green are treated for 24 hours. We found that Copaiba transiently increased AKT phosphorylations, which then returned to baseline level after 24 hours. In contrast, CBD oils steadily reduce the phosphorylation and expression level of AKT over the 24-hour period. Further downstream of AKT, we continue to observe opposite effects of Copaiba and CBD oils, where Copaiba transiently increased the phosphorylations of mTOR and P70S6K, which then returned to baseline level after 24 hours. In contrast, CBD oils steadily reduced the phosphorylation and expression level of AKT over the 24-hour period. So following the assessment of all 9 proteins within the signaling cascade, it became very clear that Copaiba upregulated in contrast, CBD oils downregulated the PI3K, AKT, mTOR signaling pathway. So we systematically apply proteomics to measure the expression and phosphorylation level of a diagnostic panels of 30 proteins, which allow us to evaluate the signaling activity of 11 signaling pathway in neurons. And we summarize our data here for your viewing. Red color indicate upregulations, blue color indicate a downregulations, compared to untreated control in white color. So as you can see, Copaiba consistently upregulated many signaling pathways in neurons. In contrast, CBD oil consistently downregulated many signaling pathways in neurons. So here is a summary of our finding that Copaiba oils increases neuronal signaling activity. In contrast, CBD oil decreases neuronal signaling activities. Copaiba induced short-term reversible responses, which is consistent with a direct interactions with neural receptors. In contrast, CBD oils induce long-term adaptive responses, which indicate an indirect interactions with neural receptors. Copaiba oils induce an anti-inflammatory response by upregulating the immunity and metabolism signaling pathway. In contrast, CBD oils induce resting states in neurons that are consistent with the anxiolytic effects. Okay? So we found that Copaiba We anticipate that Copaiba would have the strongest effects in microglial cells and lymphocytes. In contrast, CBD oil would have the strongest effects in neurons. So as you can see, Copaiba and CBD oils might have the analgesic effects, but clearly they have very different acting mechanisms and targets. There was one observation that we found very interesting with Copaiba oil, where in liver cells, we found that Copaiba downregulated the PI3K, AKT-mTOR signaling pathway. However, in neuron, we found that Copaiba upregulated the same signaling pathway. There is a logical explanation for this differential effect, because AKT isoforms have both overlapping and distinctive functions. AKT1 regulates cell growth and migration. AKT2 regulates glucose metabolism, and AKT3 regulates neuronal development. Now, in liver cells was on top, AKT3 were completely absent. However, in neurons, AKT was a dominant isoform. So the difference in the composition of the AKT isoform was the reason why Copaiba have differential effect on the same signaling pathway in liver compared to neuronal cells. But this observation indicates that the route of administration matters for Copaiba. For example, aromatic diffusion, Copaiba interact with the brain and lung tissues. For topical application, Copaiba interact with the skin tissue, and for ingestion, Copaiba interact with the GI tract and liver tissues. So it's very important that we know the effects of Copaiba on different tissue types in order to optimize its uses. So we are developing a comprehensive library that describes the composition of AKT isoform in different tissue types. We also developing an algorithm that can predict the effects of Copaiba in different tissue types, and we are experimentally testing this algorithm. We anticipate that differential effects on different tissue types is not a property unique to Copaiba, but should be applicable to other essential oils as well. So our library and our predictive algorithm would be critical in guiding the most optimal route of administration for essential oil. So what else we do for essential oil? We are developing a handheld spectrometer for this field assessment of essential oil crops, and an endoscope for real-time visualization of topical absorption of essential oils. I will sell data on this project at another time. But what I've been presenting to you is consistent with the theme of this year's convention and highlight the industry-academia interface. The collaboration between dōTERRA and Rosemont University of Health Sciences seek to understand the molecular mechanism underlying the biological activity of essential oils. In the process, we develop and apply cutting-edge technology to further our investigations. And our findings would lead to rational designs of novel products that synergize essential oils, botanicals, nutraceuticals, and pharmaceuticals. Before I stop, I would like to acknowledge Dr. Yasuyo Urizaki and Dr. Talbot for being the co-investigator on this project, Dr. Harry Rosenberg and Dr. Renee Kaufman, the co-founder of our university, for their support of biomedical research. And of course, this project would not be possible without the vision and the support of Dr. David Hill, Dr. Russia Oskildorf, and the interaction with many dedicated and talented scientists at dōTERRA, Dr. Cody Beaumont, Nicole Stevens, Alex DeBell, and Ryan Finch. I would like to acknowledge the institution that funded my research, and thank you so much for your attention.